Well, we are now on to Chapter 12, which is measurement and data collections. What we covered so far is the topic, the review of the literature, which if you remember, what is known and what is not known, which is the gap. We've looked at the purpose statements. We've figured out what the variables are. We've looked at the designs, which are descriptive and descriptive comparative, correlational, including descriptive and predictive, quasi-experimental and experimental, and we've also looked at sampling. Well, data collection is next. Well, measurement. What is it? Well, quantitative studies derive data through the measurement of research variables. Measurement can be defined as rules for assigning numbers to objects to represent quantities of attributes. The purpose of assigning numbers is to differentiate between people or objects that possess varying degrees of a critical attribute. And if you remember my slide that had the orthopedic brown shoes and my slide that had the pile of all kinds of colored shoes, we would want to put a number on the number of yellow tennis sneakers so that we could figure out how many people were wearing them, which would be of interest to a shoe manufacturer. Well, there are a few measurement theory concepts that we need to look at before we talk about data collection, etc. One is the directness of measurement, and we're going to look at direct measurement, which would include temperature and weight, and indirect measurement, which would be things like pain and depression. To measure, the researcher must first identify the object, characteristic, or element to be measured. Direct measurement occurs with concrete variables that can be measured objectively using a specific measurement strategy, such as a scale to measure weight. Examples include a lot of the data that you collect when you go out to clinical: height, weight, temperature, heart rate, and respirations. Now, indirect measurement is a process of collecting and putting a numerical value on a variable that's not so easily observed, such as stress, caring, coping, or anxiety. Another measurement theory concept that we need to deal with is measurement error. There are systematic errors and there are random errors. Now, measurement error is a part of any measurement. It's the difference between the true score and what the score was actually measured. For instance, if you get on the scale in the morning, do you really believe that the weight is true? Or do you say, well, you know what, the scale must be off a little bit, etc.? Measurement error exists for both direct and indirect measures. However, direct measures are supposed to be subject to less error than indirect measures. Now, there's the idea of random error. Random error is the difference between the measured value and the true value, but the difference is without pattern or direction. One measurement may be higher than the true value, the other may be lower than the true value, and so when you take the mean of it, it's pretty close to what the true value is. As the number of random errors increases, the precision of the estimate of the mean then decreases. Random error is something that researchers just have to put up with. Systematic error, on the other hand, is the variation in measurement that primarily moves in the same direction, compared to random, that's both up and down. Systematic error can occur because something else is being measured in addition to the concept. The most common causes of measurement error include the following. There are situational contaminants, meaning that the scores can be affected by the conditions under which they are produced. For example, the friendliness of the researcher, the anonymity of the responding situation, the time of day, the temperature of the room in which the respondents are filling out a questionnaire are examples of situational contaminant. Then there are transitory personal factors, where the scores of an individual may be influenced by fatigue, anxiety, hunger, mood, etc. Think about if you come in to take an exam, multiple choice exam, and you're very tired or you're very anxious, how do you think that is going to affect your scores? Now, there are also things called response set biases. This means that people will answer, for example, questionnaires based on social desirability or extreme responses. Now think about when we used to do the paper and pencil evaluations of instructors, and you were so tired you just wanted to get out of the class. So what you did was you picked a number on what you thought the instructor was worthy of, and you circled the same number for each of the questions. That is response set bias. Now there's also administration variation, which are alterations in the methods of collecting data from one subject to the next. For example, giving different instructions or alterations in the coding categories. Also, there's instrument clarity. If the direction for obtaining the measures are vague or poorly understood, then the scores can be affected. There's item sampling, which are errors are introduced as a result of the sampling of items used to measure an attribute. For example, a nursing student takes a test on an OB exam and scores an 89 on one test, but a 95 on a similar test because the questions were slightly different. And finally, there's instrument formats. Open-ended question may elicit different answers than closed-ended questions. And the ordering of questions can also influence the results. Well, we now want to talk about data collection. Data collection must be objective and systematic. Objective meaning that it is free from the researcher's personal biases, beliefs, values, or attitudes. Systematic means that the data must be collected in a uniform, consistent, or standard way. The success of a research study depends on the quality of the data collection methods chosen and implemented. There are 4 different data collection strategies that we are going to look at. The first one is physiologic. This involves the use of specialized equipment to determine physical and biological statuses of subjects. What separates these from other measurements in research is the use of special equipment that may require training. For example, Swan-Ganz catheter pressure readings of the chambers of the heart, blood pressure, and temperature. Advantages of using physiologic data collection methods are that they are generally objective, meaning they're without bias. They can be very precise, which means without variation. For example, if you took a sample of a CBC down to the lab and you divided it in 2 and ran that CBC in successive order, the results of that should be the same. They're also very sensitive, which means they're able to pick up very small changes. Some of the disadvantages for physiologic measures are they can be very expensive, they require special training to operate, and simply by using them, the variable measured may change. For example, someone being placed on a heart monitor may raise the heart rate because of anxiety. Another disadvantage is they may be affected by the environment. Long time ago, there used to be computers that were actually influenced by hot temperatures. So the computer rooms, which were enormous, and I was a computer operator at that time, had to be kept very cold so that it wouldn't damage the equipment. And another disadvantage is you may not be able to measure the phenomenon of interest, such as how is a physiologic tool going to measure sadness? Now, the second data collection method that we're going to look at are observations. Sometimes just observing a subject may give a more accurate picture of behavior than just asking. But the observation must be scientific and fulfill the following conditions. Number 1, the observations must be consistent with the study's objectives. There must be a standardized and systematic way of recording the data. All observations are checked and controlled, and the observations must be related to scientific theory. There are 4 types of observation. Concealment without intervention. An example of this would be if you as the researcher wanted to figure out what it was like to live in a battered women's shelter. Well, you would pose yourself as being a battered woman and go live in the shelter. And as the researcher, you're just going to kind of collect data, but you're not going to do any interventions for that population. The second type of observation is concealment with intervention. Along the same vein, you went into the battered women's shelter and took on the role of being a battered woman, but then you would maybe teach some of the women how to prevent themselves from being battered. That would be concealment with intervention. A third way is no concealment without intervention. Using our same example, you as the researcher go into the battered women's shelter, but they know you're a researcher who's looking to see how things are there, and you're not doing any interventions. The fourth way is no concealment with intervention. In this way, you as a researcher are going into the battered women's shelter. They know that you're a researcher, and you're doing some kind of intervention for them to improve their quality of life. Now remember, if the subject has not been given informed consent for the observation, the subjects must be debriefed and given the opportunity to have their data removed from the study. That's after you have completed your study. Advantages are it may be the only way to collect data, and it can provide in-depth information, and it can be very flexible. Some of the disadvantages of observational studies is that the risk of objectivity will be lost. If data are discrete, then an observational tool can be used to collect the information. If not, then field notes must be used. Now, the third way of collecting data is through interviews. Interviews involve verbal communication. Remember that. Verbal communication between the researcher and the individual in the study. The interview can be totally unstructured, in which the content is totally in control of the individual, or structured, in which the content is in the control of the researcher. For example, in my own research, the only question was, tell me about your experience of sacrificing something important. With a structured interview, the researcher would have a list of questions about the variable of sacrifice and ask only those questions. The more structured the interview, the more precisely the questions are asked, and including the order in which they are asked. Advantages are these are used extensively in qualitative and descriptive studies and facilitates obtaining in-depth information on phenomena that have not been previously studied. In general, there's a higher response rate to interviews than questionnaires. Because think about what you do with questionnaires that you get in the mail. You take them and you throw them in the circular container. Therefore, a more representative sample can be obtained. Also, information can be obtained from individuals who cannot see or write. Finally, researchers can clarify questions, which cannot be done if you receive a questionnaire in the mail. Disadvantages for interviews are it is much more costly in terms of time and money, and the sample size is limited. And there's always the threat of bias related to inconsistency of data collection from subject to subject. Questionnaires. Questionnaires are a printed self-report designed to elicit information, usually through written responses by the individual in the study. Sometimes they are referred to as surveys. The information is similar to that obtained through interviews, but the information is usually less in depth. The individual is not permitted to ask questions, and the data collector cannot ask any probing questions. Sometimes they have both open-ended and closed-ended questions. Questionnaires have the advantage of being able to be given to large numbers of individuals via the mail, but the response rate is usually a lot lower than interviews. If the response rate is less than 50%, then the representativeness of the sample is in jeopardy. Usually only 25 to 30%, and that's a pretty high number, of mail questionnaires are returned. Individually, individuals may forget to mark some of the questions, and these can threaten the validity of the instrument. The questionnaires that we've been looking at so far, I'm thinking of as yes or no answers. However, there is a questionnaire that is more precise, and these are called scales. One type of scale that I'm quite sure you're familiar with are Likert scales. These are the most common of all scales and designed to measure an attitude of a subject. It uses a declarative statement with a scale after each statement. The values are from 5 to 7 responses, with 1 being the most negative and 5 or 7 the most positive. Responses are choices that reflect agreement, evaluation, or frequency. Agreement responses are usually from strongly agree to strongly disagree. Evaluation ranges from good to bad, and the frequency from rarely to usually. These values from each item question are then summed to provide a total score. Semantic differential scales are used to measure attitudes and beliefs. The individuals are asked to select a point on a scale with 2 opposite adjectives that expresses their view of the concept being measured. A space is designed for each response, with 1 being most negative and 7 the most positive. In the instrument, the arrangement of the responses is varied to prevent individuals from choosing all of one category of response. The values are summed to obtain a global score. Now, just to reiterate some of the advantages and disadvantages. For surveys and questionnaires, advantages are they are less expensive, you get a larger, more diverse sample, it allows anonymity, and there's no bias from the interviewer. Disadvantages for surveys and questionnaires: there are questionable accuracy, there's decreased response rate, they may lack clarity, and not all can participate. With interviews, the advantages are the response rate is very high, all can participate, the interviewer knows who is answering the questions, clarification is possible, the researcher can control the order, and you can obtain more complex data. The weaknesses of interviews are accuracy. They can be very expensive. There's no anonymity, and there can be interviewer bias. This is an example of a Likert scale with a closed-ended question. How satisfied are you with your current position? This is an example of a semantic differential scale where you have the adjectives on either side of the numbers. And this slide is just a summary of what I have been talking about during this lecture on some of the attributes of questionnaires and interviews. Now, there's other ways of getting data for your research study, and this may be the use of existing data, where you use records and databases. For example, medical records, care plans, hospital records, death certificates, the United States Census, or the National Cancer Database. They are often referred to as a secondary analysis, and the researcher should state that in the article. Now, there's some disadvantages to using existing data. Institutions may be reluctant to allow researchers to have access to their records, especially if it has to do with problems within the institution, such as mortality and morbidity records of their surgeons. If the records are kept so that an individual cannot be identified, known as de-identified data, this is usually not a problem, and the hospital may allow you access. Also, the researcher has access to only those records that have survived. If the records available are not representative of all the possible records, the researcher may have a problem with bias. Some of the advantages of existing data are the data are already collected, thus eliminating subject burden and recruitment problems. Most databases contain very large populations, therefore sample size is rarely a problem, and random sampling is possible. Larger samples allow the researcher to do more sophisticated analytic procedures, and random sampling enhances generalizability of the findings, and there's potential to save significant time and money. Now we're going to kind of look at the data collection process. The first is you have to go out and recruit your subjects according to the sample criteria. But there are some problems in recruiting subjects for research because there's an increasing number of nurses doing research. Sometimes agencies are very restrictive to the use of staff to help with research activities because it costs them in money and patient satisfaction. And patients are being protected from taking part in too many investigations, and also there are a lot of HIPAA regulations. And access to patients is limited so that agencies can conduct their own research. Also, remember, you have to go out and obtain informed consent from your subjects, and then if you're doing some kind of comparison or you're doing an experimental study, you have to assign them to groups. Well, still talking about this data collection process, you have to maintain controls of treatment, measurements, methods, and setting. You also must maintain consistency in the data collection process, and you have to gather the essential data, which include demographic and measurement of any of the study variables. Finally, you have to enter the data into the computer and conduct analyses of the demographic data and the data for the study variables to address your objectives, questions, or hypotheses, and then you have to interpret the results. As you can see, all along this process, there is a lot of room for error to come in many different ways and muck up your study. And that is the end of this lecture.